Thank you to Terrapin for inviting us to talk at this wonderful congress. Really has been enjoyable so far, and certainly welcome questions at the end. I'm here today to talk about the human viral challenge studies that we do at Retroscreen. It's a decade worth of experience, and in particular, accelerating proof of concept for vaccines against respiratory viruses. Because the program also talks about the antiviral work that we do, I'll touch on that subject also. Just quite a powerful picture of the droplet spread of influenza, and bearing in mind, trying to think about exactly what we're trying to do with the influenza, RSV, and rhinovirus challenge model. So I'm going to talk about the history of the human viral challenge studies, the current experience with the model, the benefits of the challenge model, a typical vaccine challenge study, and the proof of concept vaccine and antiviral studies that we've had the privilege of working with many sponsor companies on. So looking at the virus challenge studies, they were first attempted with influenza in 1918, just after the Spanish influenza, and that was done in the UK and the US. They weren't actually quite successful in transmitting the virus. In 1946, the Common Cold Unit in Salisbury opened. Throughout the 60s and 70s, there was work done using the challenge model. In the 90s, the Common Cold Unit closed, and in 2000, these studies started at Retroscreen. Got a few pictures of the Common Cold Unit, and a bit of a story about the history, which I think is quite interesting. So it was after the conclusion of the Second World War, a new approach was pioneered by David Turrell at the Common Cold Research Unit in Salisbury. From the start in 1946, the aim of the unit was to develop methods for the cultivation and identification of viruses that cause respiratory illness. The intention was that once identified, these experimentally produced materials would be inoculated into volunteers by administering small quantities into the nose, to see if this would lead to the volunteer becoming symptomatic. Situated at the top of the hill on the southwestern outskirts of Salisbury, with open country and farmland beyond, the Common Cold Research Unit housed healthy and active volunteers in relative isolation from other people, thereby reducing the risk of their coming into contact with natural sources of infection or of passing on experimentally induced colds to the members of the general public. And this particular point is very unique to our challenge model, and it really does demonstrate the point of why it needs to be done under quarantine settings and an in-house type of model, as opposed to infecting volunteers and then go out into the community. It was a joint enterprise by the Medical Research Council, which employed the medical laboratory and office staff, and the Ministry of Health, which engaged the catering, maintenance staff, and the matron. During its time, the unit attracted 20,000 volunteers, many of whom returned at regular intervals. Very similar to our recruiting of volunteers in the present day, we do get many volunteers referring on friends, family, and sometimes coming back to be infected with other viruses. The unit was closed in 1989. This is a picture of volunteers playing cards, and they were studying transmission using fluorescein dye on the cards. So our history, a decade of human viral challenge studies. Just to take you through the slide, so it's 3 viruses: influenza, RSV, respiratory syncytial virus, and human rhinovirus. The clinical studies are divided into smaller groups of quarantines. Our largest quarantine to date is 86 volunteers. We did that in a hotel setting. Following the hotel settings, we were also using phase 1 units in and around London and Cambridge. And today, we have a new facility, which I'll talk about. So the total number of volunteers is 820 volunteers. There's never been a serious adverse event relating to the virus. And I'm glad to say that these 820 volunteers, it's been a safe and effective method of testing these vaccines and antivirals. So we call it Flu Camp. It's our own branding. Some of the volunteers came up with that name also. And this was a DNA vaccine I'll talk about in a few minutes. 86 volunteers in a hotel setting, and this is obviously post-infection, post-inoculation, and at the end of the quarantine component. You can see that's Rob Lampkin Williams. He's the Chief Scientific Officer at Retroscreen. There's the younger looking me. So that was back in 2007. The new London isolation facility, state-of-the-art, 24-bedded unit, ensuite bathrooms, really cutting edge. This is just some pictures that were taken about 6 weeks ago. We moved into the facility. Our first quarantine will take place in July, and it really is the next step of doing viral challenge studies. So what is the model? Volunteers are infected and challenged with the virus of interest to test a drug or a vaccine. They quarantine up to 16 days, and it depends, the duration depends on which virus we're using. With influenza, we can give them drugs like oseltamivir, which we helped to develop, and zanamivir towards the end of the quarantine period so that we can discharge them, and we know that the virus shedding has been reduced. HRV and RSV, we tend to keep them a little bit longer. So it's a true bridge between science and medicine to study the safety and efficacy of a new medicinal product. The key benefits, it's time and cost. So it accelerates vaccine and antiviral development, and as far as the cost is concerned, by accelerating that development, you're saving time, you're saving money, and if you're going to kill the drug, you kill it fast and quickly and cheaply. Accelerating the selection of dose and dosing regimens for later licensing studies. The key medical benefits, and as medical director, that's my priority. I serve as the principal investigator on all of our challenge studies. There's been 13 that I've served as principal investigator, and many others as co-investigator. I also sit on an ethics committee, so I know exactly what is involved in looking after the safety and well-being of these volunteers. It's 24/7 medical management. It's doctors and nurses on site the whole time. And there is the greater potential for defining better standards as it relates to this management. From the time point of viral inoculation, we can take samples from the nasopharynx, we can take blood sampling, and today we're also measuring things like cognitive function to see what effect the virus has on people, how it relates to antivirals and vaccines, to deal with the problem of absenteeism as far as the effects of these viruses on the economy and GDPs of countries like the UK and the US. And the controlled setting, unparalleled ability to gain information on cause and effect of viral transmission. We've done a viral transmission study sponsored by the UK Department of Health, and in collaboration with Nottingham University. That was interesting. We infected 9 volunteers, and then we brought in 15 volunteers a few days later to really study how this virus is transmitted, doing air sampling, foam mite sampling, and studying transmission. Greater potential for scientific insight than the traditional human field studies. It protects the community. You've got the immunocompromised groups, you've got the elderly, you've got the under 3 years of age groups, you've got those with respiratory illnesses, and not only are those exclusion criteria for allowing the volunteers to participate, but it's exclusion criteria as far as their family, their friends, the people that they come into contact with. And for healthcare professionals, medical students participating in the studies, they can't then go back into the community and work in the hospitals. And all of this has taken us the last decade to really work through, as far as infection control in the units, the exclusion criteria required for these type of studies, and really the logistics of running this type of operation. Less clinical studies to reach the safety and efficacy conclusions. The virus administration time point is absolutely known, and multiple endpoints that we can put in, obviously as primary objectives, secondary objectives. It's really quite endless. The real-time data collection and review. The samples, we've talked about the temporal relationship that they have to the time of infection. The baseline samples we can get when they actually arrive at the quarantine site. And the provision of real-time virology data to inform these key medical management decisions. And just a little bit about why it's better than field studies, besides the costs. Looking at the seasonal occurrence and the wide variation in duration and severity of disease, and the results of infection have been predictive of subsequent results in the field. These are the viruses that we work with. We can get into the details of this, and certainly if there's any questions about specific viruses, but today we've, as the slide earlier suggested, it's over 500 volunteers with influenza, over 200 volunteers with RSV, and it was one study that we did with rhinovirus, but those studies are quite simple for us. We're looking forward to, hopefully in the future, working with avian influenza, H5N1, an attenuated version, norovirus type studies, and really the setting is quite ideal for extending into these other viruses. And this is the typical vaccine challenge study flow. So first, talking about the screening, the expertise in the screening and allowing these volunteers to join a panel of volunteers. We have a few thousand volunteers on a panel whereby they arrive at the clinic, we take a serology sample, we store that for a period of 6 months, and we analyze that sample against the virus of choice that we're going to be using in the clinical study. And then we call the volunteers and find out their availability to participate. If they have low antibodies, we'll invite them to a full physical clinic, very similar to an annual physical exam, ECG, spirometry. The vaccination will take place typically 21 days prior to day 0, which is our nomenclature for the day of viral inoculation, day 0. On day -2, 2 days before inoculation, they arrive at the clinic, and that's an opportunity for my physicians to be looking out for any sign of illness. We're redoing the full physicals, the safety labs, and we're, again, ensuring that they're not going to be inoculated with a concurrent illness. So that's the vaccination phase. Coming into the quarantine phase, day 0, we inoculate, and it's really the ability every day to take sampling at different time points. Day 7 for a typical influenza study would be the day that we release the volunteers from the quarantine. We do a rapid antigen test, and they need to be symptom-free to be allowed to leave. We request them to stay in an additional 24 hours if they are still symptomatic or rapid antigen tests are positive. And then a follow-up visit a month later. This is our recruitment website. We talked about flu camp earlier. The study flow, so it's the serology sampling, the study-specific screening, then we inoculate, quarantine, and if it's influenza, we'll treat with the Tamiflu and the follow-up visit. Our screening process, the serology samples, about 70 or 80% are excluded, depending on the virus. We then have study-specific screening, that annual type visit to the general practitioner. Study-specific screening, up to 50% are lost at this point. And it could be things like the liver enzymes. We know that these viruses do affect the liver enzymes. It resolves at that day 28 visit. However, that would be an exclusion. They arrive at the quarantine, and typically out of a group of 24 volunteers, 1 or 2 may be lost to family commitments, emergencies, or perhaps they're coming in with some viral type symptoms or perhaps a fever. And then we go ahead, inoculate the evaluable subjects. This is the quarantine flow. We've talked about the quarantine day -2, day 0 is an inoculation, days 1 to 6, the ability to do all of the examinations, ECGs, the diary cards, the throat swabs, tissue collections, and on the final day, we discharge them. This is the viral inoculation, and you can see there's been a major change in how we do things from the common cold unit and to today. The change being, it's just from glass to plastic. So we use the Gilson's pipette, and this mode of administration, inoculation of virus works, and absolutely reproducible. The nasal wash procedure, And this is not your typical nasal lavage done in other settings. We've worked over 9 years to perfect it, and it is robust and it is reproducible. We use a urinary catheter. We inject a few mils of saline up into the nose, collected in the universal, and we can do this once, twice, 3 times a day in the quarantine. The data collection, the diary cards, we've got the volunteers playing PlayStation, on Facebook and Twitter, so they have time to complete diary cards, and they enjoy participating. Typically we'll do it twice a day, but we can do it 4 times a day. And I'll show you some data a little bit later that really shows the correlation between the symptoms, the physical findings, the viral shedding. We also collect tissues and measure the mucus weights. So this is a nice slide showing the symptom scores, the direct physical examinations, and the virus shedding. This was an RSV study. You can see from the duration of the study going up to day 12. Also, the total daily symptom scores. You can see that a really nice peak at the day 6, day 7. We've used plaque assay over here because RSV, the quantitative PCR, and it really does show that these 4 modalities really do show a nice curve. The titration study is done first with the virus to ensure that we have the right titer of virus going into our vaccine study or our antiviral study. And in this situation, it was Group B that was selected. It's not necessarily the highest titer of virus that we use. We're trying to see mild to moderate symptoms. We're not trying to make these volunteers as ill as possible and fevers over 40 degrees. It really is grade 1, grade 2, mild, moderate illness. There's also an effect called the von Magnus effect, which talks about a very high titer of virus doesn't necessarily bring about the most symptoms. This is just some example data from a study done in 2003 to 2004 with IDB, with a placebo, 1-dose and 2-dose regime, and that vaccine was working. Just to show the ability to get the cell counts from the nasal washes, and you can see quite clearly there the difference between the placebo, the 1-dose and the 2-dose. The tissue counts that we do, quite simple to collect all the tissues on a daily 24-hour basis in the quarantine unit. Also, a nice graph there clearly demonstrating the 2-dose impact. This is our first proof of principle that a DNA vaccination can protect human subjects from influenza. This particular study was the largest quarantine that we did. It was 86 volunteers, and it was concluded that the powder med vaccination, so it was a double-blind, randomized, placebo-controlled, parallel group study, 105 volunteers. We landed up inoculating 86 volunteers. Again, the study flow, very similar to that slide that I was showing you about the vaccination, followed by the quarantine, and then the follow-up. Some graphs looking at symptoms and viral shedding. You can see the peak at day 58, day 59, with the total grade across all subjects and physician-reported symptoms. And then on the right-hand side, looking at the viral shedding. Again, also the peak at around day 58, day 59. Colorful slide over here with all our symptoms that we collect on the subjects' diary cards. Frequency of symptoms. Again, in the study, day 58, day 59, you can see a very clear peak of symptoms. And then a slide demonstrating the vaccine efficacy. So the conclusion was that the DNA vaccine was safe and elicited immunological responses that protected human subjects from influenza. This is the first report of protection of human subjects from an infectious disease by DNA vaccination. Recently, this is an article from The Guardian in London, so a proof of concept study, a universal T-cell flu vaccine. Just some data from this particular study. You can see the vaccinated volunteers and the controls at the screen, very low T-cell responses, vaccinated, day 29, against the controls with the Wilcoxon-Signed rank test. Day 29 vaccinated are significantly higher than controls. No significant difference seen between the vaccinated and the controls at the screen. And the clinical outcome, just a nice graph there showing hours post-challenge, and you can see that the control group, more symptoms than the vaccinated group. The number of symptoms greater than grade 2, so that would be a moderate illness. Again, the severity of the flu during the challenge. And that was, so following intranasal influenza challenge, 2 out of the 11 vaccinees and 5 out of 11 control subjects developed laboratory-confirmed influenza symptoms plus the virus shedding. Symptoms of influenza were less pronounced in the vaccinated group than the controls. This provided the first demonstration of clinical efficacy of a T-cell-based influenza vaccine. Just looking at another proof of concept study that we did for Alnylam, Boston-based group against RSV, based on technology that actually won a Nobel Prize. And that was a randomized, double-blind, placebo-controlled study of an RNAi-based therapy directed against RSV. That was done in 2006, 2007. The actual project was called Gemini, and the administration of the drug resulted in a statistically significant antiviral activity with a 38% relative reduction in RSV infection rate. Another proof of concept study that we did with Biota, that was 2008, and that was a rhinovirus study. It was one cohort, 41 volunteers, and again, it achieved the clinical proof of concept. This slide is a slide that Retroscreen put together with multiple groups. Duke University were involved, and John DiVincenzo, who's an RSV expert here in the US. And what's interesting about this slide is that it's on one slide, it's comparing influenza and RSV. It's looking at the viral load and RSV symptoms, and it really does show what the model is capable of looking at. The message from this particular slide is really the duration of shedding with RSV. And I know, again, we're looking at antivirals with this particular picture, so not necessarily vaccines, but the duration allows for antiviral therapy over a longer period of time, whereas with influenza, you've got a small window of opportunity. So certainly an opportunity for further development in that area. Some key research, as I mentioned, with Duke University. This is sponsored by the Defense Advanced Research Projects Agency, that's DARPA, and we've had a 3-year collaboration now with Duke, and we've published some papers, and it's gene expression signatures diagnosing influenza and other symptomatic RSV viral infections in humans. And essentially, with this novel diagnostic, we want to be able to diagnose respiratory viral illness 24 hours, 12 hours prior to symptoms actually occur. And you can see the benefits of a diagnostic like this, why the military would be interested in it, not deploying their troops if there's an outbreak. And obviously with an epidemic or a pandemic, the usefulness of this type of technology in a city like New York or London. And this is the team, medical team and nurses, support staff. And on this slide, I really want to talk about the correlates of protection that this type of model can actually offer. It may not be for a vaccine that the vaccine is actually producing antibodies that can actually show efficacy. And yesterday in the panel discussion, we were talking about the regulatory agencies, and I think the future is really working with the regulatory agencies to understand the model and to see what else correlates to the protection against these viruses. We're talking about the T-cell responses, we're talking about nasal IgA and other mucosal responses, other cytokine responses. This model allows us to learn about it, and it really has been a privilege working with fantastic groups, researchers around the world, particularly my time in the UK. And with the US researchers, it's not only Duke University, it allows us to further explore and investigate this key area. I'd like to thank, again, the organizers, but I think most importantly, I'd like to thank the volunteers who have dedicated weeks and months of their lives. It's interesting meeting these people and finding out why they want to participate. And I've taken the consents, and I need to ensure that they understand exactly what they're getting into. And for me to be able to say to them that if I was to choose a particular clinical study to myself volunteer for, this is the type of study that I'd be interested in volunteering for. We know what we're getting into with these viruses, and these are not first-in-man studies. So by the time we get to our challenge model, we really do have a safety signal. And on that note, if there are any questions, I'd be happy to answer them.
So we have time for one or two questions. Go on.
Really nice studies. I'm wondering about the elderly population where flu vaccine efficacy has once more become fairly controversial. Do you do any studies in a high-risk group like that? Or think about it?
It's an excellent question, and because I had 44 slides, I didn't want to put a 45th slide about the future development. But we're looking at those at-risk groups. We're looking at asthma and COPD. We're looking at collaborations with academics within the UK who are interested in these populations also. And certainly it's my belief that it is possible. In a controlled setting, my father's 65 and there's absolutely no reason why he wouldn't be able to handle a influenza, RSV or rhinovirus study. The exclusion criteria would be key. And certainly for asthmatics, the months go by in the winter, and the asthmatics, obviously we would be selecting them with very close monitoring of spirometry. There's no reason why we can't start looking at this model. But at the moment, there is enough work for us to do in the antiviral and vaccine fields of the healthy volunteers. Retroscreen is very interested in moving in that direction. Thank you for the question.
Any other questions? Well, let's thank Anthony again.
Thank you.